as possible, depending on the limited time at the end of the session. So, but if we don't get to all the questions, just know that we are collating all the questions and we'll have them all in a document for you and answer to you and send in the post-webinar email. Also, at the end of the session, so after the questions, there'll be a small survey that will pop up. So we'd really appreciate if you could take the time to complete it. It's very short, only 5 questions. It'll just help us to improve and continue these education opportunities and give us ideas for you guys as healthcare professionals on what you would like more information on. So I now want to welcome our presenter, Teresa Mitchell-Paterson, a naturopath, a qualified nutritionist, and a Master of Health Science in Human Nutrition with over 20 years experience in research and practice. So welcome, Teresa, and thank you so much for your time today to present on a topic that I know is on the minds of so many health professionals. We have heard a lot that olive leaf extract is used for immunity. It'll be finally good to see where the evidence sits. So I will now pass over to you.
Hello, good afternoon. I don't know if any of you are in Sydney at the moment, but it's definitely wild and woolly out there. I've had a lot of fun researching olive leaf extract, and today we're going to have a look at some of the very exciting things that we can use olive leaf extract for, and more on the terms of what it's specifically good for. So I'd like to just share a little bit of my experience with olive leaf extract with you. It's not a great deal, but it kind of fits in with the research. Some 15, 20 years ago, you might have found that there was a bit of a surge of interest in olive leaf extract, and they were suggesting that it was good for all sorts of things. And I used it for a patient of mine that had a tooth abscess. Now, normally when I look at an acute condition, I tend to double the dose of whatever I'm using for a couple of days just to make sure that there's a high plasma level, the active ingredients. And that's what I did. I used 2 tablets 4 times a day for the tooth abscess. This lady had tried some antibiotics prior to me having some influence on the abscess, and the antibiotics weren't working, and the olive leaf extract did in actual fact take the tooth abscess down enough for her to go and have some fillings on the tooth. And also recently I've started using olive leaf extract again, particularly in an elderly gentleman who has a pacemaker and he's got fibrosis and bronchiectasis. It hasn't cured the bronchiectasis or the fibrosis, of course, but what it does do is it keeps the level of mucus down, and it's kept him well, because you can appreciate that with bronchiectasis, it could be quite devastating to have, a cold or a flu. So we're going to look at the empirical use of olive leaf extract and the current evidence, and also the possible mechanistic actions. So having a bit more of a look at that. And also the strengths and limitations of all the current research and any further research that needs to be done for any of you that are interested in some extra research subjects. So, olive leaf extract, one of the oldest trees on Earth, the olive leaf, dates back to over 7,000 years ago, and archaeological reports in the Mediterranean state that olives were cultivated for Minoan civilization in 3000 BC, so that's almost 6,005 years ago now. And olive oil was used for body health and gastrointestinal conditions in the skin, actually, well, mostly for everything. And we've since then started to look at cultivation of the olive plant itself, and lately looking at cultivation of the olive leaf. And one of the earliest recollections of using olive leaf was from the Spanish conquistadors. They were looking at olive leaf for various illnesses, although they didn't have a lot of written evidence. The aqueous extract was used for mostly the treatment of gout in the Mediterranean area. It turns out that olive leaf extract has an enzyme that inhibits the activity of purines. This is based on ethno-botanical knowledge, but in the research that I've put there on that particular page by Hashmi, he quotes Fleming et al., and they investigated the possible inhibitory effects of the 80% ethanolic dry olive leaf extract against a drug allopurinol, which I'm sure quite a few of you know is for gout. And they found that there were 9 isolated compounds in olive leaf extract, several of them phenolics, that inhibited the xanthine oxidase, which is the enzyme which causes gout. So that was quite an exciting piece of study. Other documentation, but not formal. This is ethno-botanical at this stage. Olive leaf extract was used as a febrifuge in the Spanish War in the 1800s, and it was also used for colic. And Maud Grieve, in A Modern Herbal, gives mention of the parts used. She doesn't actually go into any great depth about olive leaf extract, but she does mention that the parts used are just the fruit, the leaf, and also the bark. It's also mentioned in the French Agence de Médicaments, and it's enjoyed a long-term traditional use. And in Argentina, it was used for diarrhea, respiratory, and urinary tract infections. I am very sorry. Thank you for putting me right there. Okay. The first formal medical mention of olive leaf is an account by Daniel Hambury 150 years ago. And it describes the ability of olive leaf extract to cure cases of severe fever and malaria. And that was published in the first pharmaceutical journal of the time. And the author wrote that he discovered the efficacy of a tincture around about 1843. It's a rather interesting little recipe, and I'm quoting from him at the moment. Boil a handful of leaves in a quart of water down to half its original volume, and then administer the liquid in amount of a wine glass every 3 to 4 hours until the fever is cured. In that, I would note he probably had a reasonably high amount of the olive leaf being precipitated into that liquid. Hambury thought it was likely the bitter substance, later known as a bitter glycoside, in the olive leaf that conferred its success. And later on, we discovered that this bitter principle was polyeuropean. And this is one of the most researched compounds in the olive leaf extract itself. It's a powerful polyphenol antioxidant, and one of our traditional integrative practitioners, some of you I'm sure have read Hoffman, Dr. Ronald Hoffman, MD. He practiced medicine for about 30 years in New York, and he used olive leaf extract quite frequently. And he mentioned in several of his books that it was used for a broad variety of health conditions, including conditions related to viruses, retroviruses, bacterium, and also protozoa. And that's going just on his clinical studies, in case studies only. Later on, there was some early trials, and in the early 1900s, the Upjohn Pharmaceutical Company identified calcium alenolate, and this was tested in vitro. So we're talking about petri dish testing at this stage, and it demonstrated a high capability for viral inhibition. Unfortunately, it didn't translate to human trial. In an uncontrolled in vivo research in animal and human, they identified that the compound bound to proteins in the blood serum and made it ineffective. Later on, East Park Research identified the exact reasoning why calcium alenolate did not transfer to in vivo success. The compound actually needed a slightly altered chemical state. And this was when they patented alenolic acid, or D-lenolate, and they registered and trademarked it. At the time, it was used as a broad-spectrum antimicrobial and also to boost human immune function. There isn't any sort of large study on it, however. It's all case studies. In clinical practice, several doctors used the supplement, found it quite impressive, and Dr. Bernie Mizok conducted a double-blind study on 30 subjects for 60 days and found that it reduced candida symptomatology. And I've actually started using olive leaf extract for that precise reason, but stay tuned because I've only just started using it. So hopefully I'll be able to disseminate some case studies once that's complete. So a summary of the studies from 1980 to present. To date, there is not a huge amount of evidence. It is largely in vitro, animal and human, and in vivo, animal only, and one human trial. And we're going to talk about that human trial later in the piece. It was quite a small human sample, very small population base. Treatment fidelity could be an issue, as the amount of olive leaf extract in the recent research is not discussed in clinically applicable terms for us to use, particularly not in human use in vivo or in animals in vivo. And no specification of the amount of actives in the olive leaf extract are constant. So we've still got a fair way to go on the evidence. However, there's some pretty convincing scientific understanding of olive leaf extract and how perhaps it marries up with the ethno-botanical use of olive leaf extract. Phytochemical research carried out on olive leaf extract has led to the isolation of flavonoids, flavone glycosides, flavonones, iridoids, iridane glycosides, saccharidoids, and saccharidoid glycosides, triterpenes, biphenyls, benzoic acid derivatives, xylitol, sterols, sugars, and a few other types of secondary metabolites that we haven't even looked at or haven't even been experimented with at this stage. Of those chemicals listed on slide 8, very few components have really been in depth and explored, except D-lenolate and a focus on polyeuropean. The leaf also contains hydroxytyrosol, which has been stated to be almost as antimicrobial as polyeuropean. And the olive leaf is a remarkable source of bioactive phenolic compounds containing a minimum of 5% polyeuropean on a dry matter basis. The exact mechanism of the antimicrobial activity of polyeuropean is still not completely established. We're still finding out some new research on it, some new possible pathways. So these are postulated at best, but possibly early research suggests that there's a direct stimulation of phagocytosis as a response of the immune system to microbes of all types. It's quite broad spectrum, and I'll talk a bit more about that. Some authors have proposed that it is due to its ability to penetrate the cell membrane of a bacteria and get to the target site, and that interferes with the amino acids that are necessary for the growth of specific microorganisms. And in viral in vivo study, one of the suspected targets for olive leaf extract is an attack on the surface glycoprotein subunit of a virus wall, and that prevents replication. In a later study by Chemichem and also Can, it stated that the polysaccharides and the sugars in olive leaf extract also have potential antimicrobial effects as well. So, as I mentioned, there's still a few parts of the olive leaf that we need to explore and experiment with. Olive leaf extract may be beneficial in Salmonella enterica, Micrococcus luteus, and has a moderate effect on Escherichia coli and Bacillus cereus and Enterobacter. But again, we need further studies in vivo. They suggest there could be potent in vivo characteristics from the olive leaf extract. So according to the studies, the bioactive biophenols and flavonoids use different mechanisms to exert their beneficial effect, which may explain their additive effect. De Bock and Thorstensen and Derek suggest several studies demonstrate that the phenolic compound from olive leaf extract may be synergistic when used in the same proportion as found in the natural leaf. It's been found that in vivo in mice, the polyphenols and their metabolites were absorbed, metabolized, and present in the plasma, the heart, the kidneys, the testicles, and the blood-brain barrier. And for me, that's leading to the thought that the olive leaf extract can potentially reach many areas of animal tissue. And it's interesting that we also find that although it hasn't been found in actual tissue, it does spread into the plasma in human trials. So looking at the phenolic compounds derived from the olive leaf extract, particularly the hydroxytyrosol and the polyeuropean, taken either as a liquid or a capsule, De Bock looked at quantifying the bioavailability and metabolism when taking olive leaf extract. Now, I must state that it wasn't a big study, but they had 9 volunteers, 5 males, 4 females, and their ages were between 42 and 50. and they were randomized to receive either a capsulated or a liquid olive leaf extract. Plasma and urine samples were collected at fixed intervals for 24 hours post-ingestion. And firstly, the conjugated metabolites of hydroxytyrosol were noted as metabolizing the fastest. And there was no difference in the absorption of hydroxytyrosol with tablet or liquid. But peak European concentrations were found after ingestion of liquid rather than capsule preparations. Now, that doesn't mean to say that the capsule doesn't work. It just means to say that the liquid works faster than the capsule. And that kind of makes sense because in an olive leaf extract, you're going to have some alcohol, and alcohol does cross into across the stomach barrier much more rapidly than a non-alcoholic preparation. So the conclusion was that olive leaf extract effectively delivers polyeuropean and hydroxytyrosol from the leaf metabolites into plasma of humans. And olive leaf extract appears to demonstrate systemic bacterial effects because it could potentially have that because it spreads everywhere in the plasma. And if you look at some of the other evidence, that kind of makes sense because there have been some trials in vitro, in cancer cells, in cardiovascular tissue, in the immune system, in respiratory, gastrodental, and other chronic diseases. And we're looking currently at how the exact effect is being elucidated. We know from a naturopathic point of view, those of us that are naturopaths, that treating the system to bring about homeostasis is to use herbs that can affect internal organs and also reach difficult places such as the skin, etc. And it would seem that being in the plasma, olive leaf extract would be a promising extract to use to reach very distant sites. So there's a term called phytoalexin. Now, the term phytoalexin was coined by Mueller, and it's on the basis that a plant antimicrobial compound can resist disease. And it's synthesized and accumulated in plants after exposure to microorganisms. And he stated that they had a potential benefit to human immune systems. And phytoalexins display an enormous diversity belonging to various chemical families, such as those found in the biophenols of olive leaf extracts, in the isoflavones, the flavones, and many of the other plant chemicals. Now, phytoalexin was then sort of superseded by another term. So if you come across this term, it's quite an interesting term. Phytoanticipin. Phytoanticipin is a chemical that basically anticipates when to bind together. So polyeuropean and ligstroside, when they should bind together to resist disease. So it's only going to happen after there's been an exposure to an antimicrobial. So it almost anticipates or waits in the plant, and it's ready to group together to make a chemical in the plant more effective as an antimicrobial. So what we know in vitro studies that we want to put across that in vitro there have been some very specific testing on some of these diseases such as tuberculosis, typhoid fever. And typhoid fever affects roughly 17 million people annually, causing over 600,000 deaths. So it would certainly be worth doing a trial in typhoid fever. Vibrio parahemolyticus infection. This is not a disease that is transmittable from human to human, but it's a really horrible gastrointestinal tract disease causing blood in diarrhea, nausea, vomiting, headache, and fever, etc. And it's acquired by eating raw and undercooked shellfish or drinking contaminated water. I'm sure everybody's heard of Streptococcus aureus, and it's suggested that olive leaf extract could have potential use in penicillin-resistant strains. Klebsiella pneumoniae, it's rare, but it can be seen in immune-compromised patients after long-term antibiotic use and immune-compromised patients, and also Plasmodium falciparum, which is malaria, and E. coli, and I'm sure those of you that are practitioners, you would have come across E. coli in bowel conditions a number of times. So certainly worth looking at. Later on, there was a study that was used to test olive leaf extract in oral microorganisms, and that fits in with my earlier little story, which I was quite happy to see when I did the research of the abscess underneath a lady's tooth many years ago when olive leaf enjoyed a bit of a resurgence. So olive leaf extract can treat biofilm-related oral diseases that are often correlated with antibiotic resistance. And there's a possibility that plant-derived antimicrobial agents could enhance existing treatment options. They might be able to be used side-by-side antibiotics. Therefore, the rationale of the present report was to examine the antimicrobial impact of several natural extracts on oral microorganisms. So they did test some other natural antimicrobials, but olive leaf extract fared very well. It was effective against at least 10 different types of oral microorganisms and one Candida albicans strain. And it was both able to inhibit and also reduce the amount of bacteria in the mouth from replicating. So some quite positive research there. In vitro viral specificity, herpes, mononucleosis, some hepatitis virus, rotavirus, and the respiratory viruses. And theoretically, it has some effect on HIV, so more testing needs to be done for that, but research is looking promising. So, summary of the hypothesis of mechanism of the action on antiviral. It may interfere with the food chain of amino acids for viruses. Buna suggests that the polyphenols can also down-regulate cytokine storm and protect the mucosal tissue of the lungs. So quite a few of the really nasty flus that are going around that affect mainly children and also the aged have a very potent cytokine storm. So olive leaf extract may be very beneficial in assisting those types of flus and viruses. Can interfere with the food chain, I've mentioned that. contains the virus by inactivating and preventing budding, shedding, and assembly, penetrates the viral shell, prevents replication, and in some viruses, in retroviruses, it actually neutralizes the reverse transcriptase and protease, and also pumps up phagocytosis and gets those white blood cells working well. So this slide is just to sort of suggest that some parts of the plant are more effective than others. And this paper is again by Hashmi, and also some discussion from the alternative medicine monograph. And it's suggesting that the different chemical compounds in the olive leaf extract demonstrate different activity and a different level of activity, and also a different level of potency. In some human immune cells, some human immune cells actually express a higher polyphenol receptor, and that allows them to uptake the polyphenols from olive leaf extract a little more quickly, and also to regulate tissue damage. So the action of uptake initiates signaling pathways to commence immune responses, and it may also activate epigenetic changes. In a study on mast cells, and this was in purified peritoneal mast cells, obviously not human, they were then treated with hydroxytyrosol or polyeuropean, and also there was a comparative study performed. The reaction was measured for mast cell activation, and biochemical and morphological findings of this study showed that for the first time that the hydroxytyrosol and the polyeuropean inhibit mast cell degranulation. So polyphenols can regulate mucosal immune response in the gastrointestinal tract, allergic disease, and also demonstrate anti-tumor immunity. So some quite positive research. I like this little picture from Margone. I like it because it sort of explains what could possibly happen from a pharmacodynamic immune modulation point of view. So Margone et al. Researched the modification of healthy human immune responses of olive leaf extract via their expression of T helper cells. And it also increased interferon production. So the action of interferon induces higher levels of T cytotoxic cells, the CD8s, and the natural killer cell activation. And olive leaf balanced interleukin 10 from Treg cells, and therefore balanced interleukin 17, which is a not so good one if we want to put it in simple terms. And that's actually paramount to prevent autoimmune and allergic disease. So this, in fact, the olive leaf extract pharmacodynamics could, in fact, reduce ongoing inflammation. That's probably where that reduction of cytokine storm comes in. And interleukin 17 is mostly found in the mucosal cells, which may hint to why olive leaf extract is more effective in the respiratory tract and the gastrointestinal tract. And another added benefit was that it also reduced COX-2 in human immune cells, and also increased the nitric oxide in macrophages to increase mucosal protection. And it suggested that olive leaf extract is a regulator of the T cell subsets. So some quite positive outcomes from that particular study. So it isn't just about its primary pharmacodynamics, it's also about its secondary pharmacodynamics, because the innate immune system is the body's first line of defense to sense and eliminate invading pathogens. And during an infection, the host's inflammatory processes mediate the destruction of microbes. If inflammation is upregulated, then this can lead to increased tissue damage and other pathophysiological disorders such as sepsis. And olive leaf extract demonstrates a secondary immune response by downregulating nuclear factor erythroid 2, or NRF as we know it, and reduces inflammation. So a couple of different pathways here where we're getting added benefits. And additionally, both the fruit but also the leaf are antioxidant and antimicrobial. The term antioxidant is related to the use of the plant chemical that manages the reactive species. And I know that most of you would understand that, but just to reiterate, the antioxidant in olive leaf extract manages reactive oxygen species and nitrogen species to assist several body functions. So here's the holistic approach here. And also energy supply to cells. It can assist with detoxification via liver sulfation, glucuronidation, etc. It improves chemical signaling, immune function. We know that reactive oxygen and nitrogen species are continuously produced in the human body, and that it's carefully controlled. But the action of the polyphenols can go a long way to assisting superoxide dismutase, glutathione peroxidase, and also catalase. And if we've got too many reactive species via an exposure to an oxidant substance like a microbial infection, then we've got failure of the defense mechanisms, and therefore damage to the biomolecules like the DNA, the lipids and proteins, etc. And therefore that affects the cells themselves and causes what we call a gene toxicity or genotoxicity. And olive leaf extract goes some way to moderating the damage of reactive species. So another additional thought. In the one and only Cochrane randomized controlled trial, people are asking questions about common colds, and I think hopefully this might help. 32 high school students who played sport for an elite team at their school were recruited to a randomized controlled trial. It was quite an interesting little report because if you read it, and hopefully you do, because there's lots of interesting comments in it. The dietary intake of the athletes were suboptimal. So what they're saying is that the diets of these school children aged 16 to 19 would not have been stimulating the immune system whatsoever. So there was no external help given here. And the students were not, that participated in this particular controlled trial, were not allowed to or not have been able to take any other polyphenols like quercetin, propolis, turmeric, curcumin, etc. in the previous 3 months, but there was no danger of that happening. They were each allocated olive leaf extract containing about 100 milligrams of Oli European or a placebo supplementation for a period of 9 weeks. during their competitive season. And they were measured twice weekly for illness and respiratory microbes, and their training load and well-being scores were analysed by Sporting Upper Respiratory Illness Questionnaire, or SUPRES, during their training. And the study conducted suggests that although the athletes had a similar incidence of illness, there was a significant 28% reduction in sick days in the olive leaf cohort. So the supplementation over the season did help to reduce upper respiratory tract length of disease and ability for the student to return to play. So yeah, obviously we need some more trials, but that was quite interesting. Somerville suggests that the mechanism of action was that firstly, oleuropein acts as an antioxidant with a dose-dependent inhibition of the copper sulfate-induced oxidation of the lipoproteins in cells, and that it increased nitric oxide production in macrophage. And what that does is stimulate the little white blood cells to whiz around, if you like, and start doing up the doing all the mopping up, and it increases their functional activity. And oleuropein also had an effect on the etiology of the upper respiratory tract infections by producing some strong antimicrobial effect against gram-positive and gram-negative bacterial infection, and antiviral against the respiratory syncytial virus, or RSV, which is a very common upper respiratory tract infection. And an interesting little case study, but I do urge you to write case studies. If you are using olive leaf extract, please send in any case reports that you do. They don't have to have a massive literature review added to them, because basically this will inform a literature review. But if you do have a case study that you'd like to put forward, it's always interesting to hear about it. Useful in a patient that had herpes genitalis, quite a difficult condition to treat, but there was nil occurrence of the disease for 12 months whilst the patient was on the olive leaf extract, and that's by a chiropractic doctor. This is just a notation. There is some suggestion that oleuropein is among the herbal constituents that act as an inhibitor of CYP450 enzymes, similar to that of capsaicin from chili, and glabridin from licorice, and resveratrol from red wine. So there's been some suggestion that there's a herb-drug interaction, but very little interactions have been actually reported. So clinical uses based on in vitro studies, decreased time to recovery, so quicker recovery, used for infection of many different types of bacteria and viruses, an immune modulation effect, a viral modulation effect, and additionally a tissue stabilizer. Safety in patient counselling. Unfortunately, there's not a great deal of evidence there. There's no data for safety in pregnancy and breastfeeding, so best not to use it, and no data for infant use. There's a possible hypersensitivity to the olive plant family. I've never come across this myself in practice in nearly 30 years, but if you have, obviously it's one to avoid. Possible caution with diuretics and renal disorders. In vitro suggests a possible caution with blood thinners, but that has not been proven. in vivo. So in general, the safety reporting from European Medicines Agency, adverse events are seen in only 5% of studies. It possibly could cause a little bit of coughing and vertigo, a bit of muscle discomfort and headache. I would suggest if that was happening that you reduce the dose immediately and or cease the dose depending on the severity. No serious drug-related reports or moderate adverse events. And we do have an ethno-botanical reported use for over 30 years with no safety issues. So suggested dose and use is widely varied. I suggest you go with the manufacturer's use. If you have a look at the Wellgrove product, it does suggest that it's 15 mils per day. I have used it as much as 7 mils per day. 7 mils 3 times a day in acute, and then drop down to 15 mils a day. For infections, safe in 2 to 4, possibly 8 weeks, possibly safe for 12 months. I haven't got to that stage yet. So, promising activity requiring future human research. There's plenty of opportunity out there if you want to do some research on olive leaf extract. We need some pure isolation studies on individual and compound components of the olive leaf extract. We need some liquid extracts with dosing efficacy, hydroxytyrosol versus the European antiviral activity in vivo with a large cohort if at all possible, standardization suggestions, and in vivo random controlled trials, prospective and also retro, if possible, larger cohorts, longer time frames, if possible, the gold standard of 6 months. And don't ever forget that case reports and or studies are highly valuable because it helps the rest of our fraternity to look at what's possible with olive leaf extract. And I'm now happy to answer some questions.
Thanks so much, Teresa, for a super informative webinar. We did have a few questions come through. The first one is, might the immune supporting benefits of olive leaf extract be useful in those using immune modulating drugs in rheumatoid arthritis and psoriasis?
It would be safe to suggest, from what we know, that it could be safely used alongside it. You'd have to let a practitioner know, whoever's prescribed the medication that you're going to use olive leaf extract. But because we haven't got any safety data, we must be aware of the fact that there is potential for it to inhibit the CYP450 enzyme, but in a very small dose, I think it should potentially be safe.
Thank you. The next one, is there any research for use in treating the common cold?
Not in vivo.
No.
There's plenty of evidence in vitro for gram-negative, gram-positive bacteria that may actually incite a common cold. However, there's nothing in vivo.
Yeah, thank you. The next one was, do you have any thoughts about olive leaf extract prepared with ethanol versus glycerol?
There's no evidence to suggest that the glycerol has an effect. So the evidence, the trials that have been done so far have all been on ethanol extract. So that's yet to be elucidated.
Okay. Should the initiation of olive leaf extract be gradual?
I like to do that. For those of you that are practitioners out there, it's always a good idea to try take your dose and to introduce things very slowly. Some patients are extremely sensitive, some are not sensitive at all. If you know your patient well and they're sort of a person that does well on what I call horse doses, then by all means start at the upper end of the prescribing spectrum. But if you've got a patient that is very sensitive, then you could even start with a drop dose. I'm not adverse to that. So titrating dose up is always a good option.
Yeah. Thanks, Teresa. We do have one more. Is there any information you have about treatment of asthma?
I don't have any, I haven't seen any studies on asthma, unfortunately, at this time. What I would say is that it potentially be preventative for the occurrence of an antimicrobial or a viral attack. And we all know that when a patient has asthma and they get sick, it's much worse for them.
Thank you. Another one has popped through. Would you, Teresa, advise olive leaf extract for a cold with the current research?
Definitely, definitely would. In fact, now that I've done this research, it's popped back into my memory. I did use it quite a lot. I don't know if any of you are old enough to remember the 1995. Some of you might not have been born then, but there was quite a resurgence of olive leaf at that time, and we definitely used it for colds and flus then, and I'm using it for colds and flus now more as a preventative, because as I mentioned, my gentleman with the fibrosis and bronchiectasis, the last thing I need is for him to get a cold or a flu. But I have put a number of people on it now and watch this space. Hopefully they won't get as many colds or flus because they are immune compromised patients. But I would certainly use it on the onset of a cold or flu, basing that on the in vitro information.
Yeah. Are you, sorry, I've just got it. There's another one that come through. Would you use olive leaf extract alone or in addition with other supplements? That was my exact question, so thank you.
Yeah, I think we're prone to overdoing it. We sort of, somebody gets a cold or a flu and we throw vitamin C, zinc, a herb, all sorts of additional products at them. I think use one herb and use it well. So I'm not adverse to using one thing.
Yeah, awesome. Thank you so much, Teresa. So that is it for the questions. So thank you so much again, and just letting everyone know this webinar has been recorded, so it will be available on the Olive Wellness Institute website, and we will send out a post-webinar email with those questions that Teresa has just answered. So I will leave you now with the survey, if you could please complete, just a short one. And yeah, thank you everyone for joining us. Thanks, Teresa.